Good afternoon, everyone. I'm Haley Honeycutt. This is my Genetics 311 video for Exam 3, Part 2, and I'm doing Prompt 32B. So today I'll be talking about the experiments and key findings of the genetic code. For a little background information, these are nucleotide bases for RNA, A, G, U, and C, and we'll be talking about these and referring to these throughout the video. You'll also need to know what the genetic code is. The genetic code is made up of RNA and DNA molecules, which use sets of nucleotides to transfer genetic information throughout living cells. Now let's talk about acid, dude. In order to provide for all 20 amino acids, we'll need a 3-letter codon that does not overlap. So 4 to the power of 3 gives you 20 amino acids. So in 1961, it was determined that in order for RNA to provide the template for protein formation, we need 3 base non-overlapping codes. So now the main question is, which codon codes for which amino acid? This is where Nirenberg and Mattaei come in to do the first experiment in regards to this question. Side note, he looked good. So what these fine men did, they took a poly-U, or long string of U's, mixed them with each amino acid individually, and then they radioactively labeled amino acids. If the protein is radioactive, then the labeled amino acid must be within the protein, and therefore the codon codes for that specific amino acid. So what this means is that a specific RNA sequence is coded for a specific amino acid. So they found out that U was phenylalanine, triple C's are proline, U's are lysine, and they never could get G's to work. So after that, Nirenberg said, Mattaei who, and went and partnered up with Linder. So what these two did was a little bit of a different experiment than before. They performed what was called a triple binding assay. They took a trinucleotide, like AGC or CGA for example. They mixed that with a ribosome and with each radioactive amino acid individually. This experiment allowed for binding to occur as well as filtering out the unbonded amino acids. After this, they would have to check to see if the filter paper was radioactive or not. This allowed them to find every single codon, coming to a total count of 64 codons. They then repeated the process, but then they only found 61 codons. Do you know why? It's because the other 3 codons are stop codons. 1, 2, 3. So now we know that there's a total of 64 codons and 3 stop codons. Thanks for watching, and I hope to see you all next time.